Dear students, in this module, we'll be continuing with our ongoing chapter, Chapter 4, Animal Kingdom. Firstly, we'll have a glance at the 10 different phylum included in the kingdom Animalia. So, the kingdom Animalia, on the basis of level of organization, symmetry, the body cavity or the coelom, that's the hollow body cavity between the gut wall and the body wall. It can be classified into different phylum. The most underdeveloped phylum among the kingdom Animalia is the Porifera. Poriferans are organisms which have cellular level of organization. Their body cannot be divided into two equal halves, therefore asymmetrical. They lack a true coelomic cavity, hence a coelomate. On the basis of the level of organization, the organisms belonging to the various other phylum, they may have, because they are more well developed, they may have a tissue, organ or organ system level of organization. Talking about the Coelenterata, the next phylum, or the Cnidarians, they exhibit radial symmetry. They are acoelomate organisms. The same goes for Ctenophora. The other organisms, that is the flatworms, Esculmenthes, that is the roundworms, Annelida, Arthropoda, mollusca, echinoderms, hemichordata and chordates, they have either organ or organ system level of organization. Some of them are acoelomate, like Platyhelminthes, that is flatworms. Esculmenthes, they are pseudocoelomate, whereas the phylum from Annelida to Chordata includes coelomate organisms. Talking about the phylum Porifera. The phylum Porifera, the organisms have been grouped under this phylum on the basis of, firstly, the level of organization. Second, the criteria is the body symmetry. The germinal layers, that is the ectoderm, endoderm, mesoderm. The coelomic cavity, which is the hollow cavity which is present between the gut and the body wall. The habitat, whether the respiratory, digestive, circulatory system, whether they are present or whether they are absent. The mode of reproduction, certain unique features like the water canal system, and the examples of poriferans. Firstly, talking about the general characters. Poriferans exhibit cellular level of organization. They have cell aggregate. They form cell aggregate. They call this poriferans, that refers to pore-bearing organisms, because they do not have a well-developed, or they do not have organ or organ system. By the process of osmosis and diffusion, they'll carry out the respiration, digestion and excretion activity through a single pore, that is the osculum. The symmetry, they are asymmetrical. Some may exhibit radial symmetry. They do not have a hollow cavity, therefore referred to as acoelomate organisms. Moving on to the next characteristic, that is the habitat. Generally, we can see that they are found in the ocean bed, the seabeds and the ocean trenches. And therefore, their habitat is aquatic, generally marine. They could be attached to the surface because they are sessile, they are non-motile. They could be solitary, that is alone, or they could be found in colonies like the bacteria, they are present in colonies. They are diploblastic organisms, that means they've got two layers, outer ectoderm and inner endoderm. The organ system is not developed, therefore the digestive system is absent. The digestion will take place inside the cell This opening which we can see over here, that is what is referred to as osculum, and there is also presence of oscular fringe. Respiratory circulatory system is absent because they've got a cellular level of organization as of yet. Talking about the reproduction, they exhibit a sexual method of reproduction which takes place through fragmentation. The sexual method involves the release of sperm which fuses with the egg as shown in the purple colored diagrammatic representation that results in the formation of larvae. Larvae formation takes place and therefore the development is indirect over here. With the help of water current, the larvae, it will move because they do not have the organs for motility, and they'll rest on a hard surface and develop into an adult sponge. This is how the sexual reproduction takes place in case of sponges. They are generally hermaphrodite, that means bisexual organisms. Fertilization takes place inside the body of a sponge and therefore it is internal, and development is indirect. Talking about the unique feature of the poriferans, they've got the presence of a water canal system. The porifera, as the name suggests, they've got many pores throughout their body, which are referred to as the ostia. So through these pores, the water, along with the food particles, it moves in the cavity, which is referred to as the spongocoel. The digestion, which is intracellular, takes place, and the excretion takes place with the movement of water through the osculum. So the water moves in through the ostia by the process of osmosis. Movement takes place through the spongocoel. The outward movement is mediated by osculum. The canal system is used for gathering of food, for gaseous exchange, as well as for the removal of waste, as it does not have the organ system. Talking about the cells, basically, the body of sponges has got three layers. The pinacoderm, it is made up of these cells called as pinacocytes and porocytes. The mesenchyme, which is made up of amoebocytes, and the choanoderm, which is the innermost layer, which is made up of choanocytes. When we talk about the cells, that is the amoebocyte, they include various kinds of cell which are involved, like archaeocytes, which are totipotent cells. Amoebocyte, they also facilitate the process of phagocytosis of the food particles. The choanocytes, it can be seen in the diagrammatic representation, are the collar cells. In the diagram is the spicules, is exhibited the presence of spicules. The internal skeleton of sponges is made up of spongin fibers or spicules, or both. The spicules could be made up of calcium carbonate or they could be made up of silica. Sponges, they are filter feeders like cycon or spongilla, euspongia, etc. The body of the sponges is supported by spongin fibers and the spicules. So the spicules, they've got glass-like pointed thorny structures. The amoeboid cells, they help in phagocytosis. The collar cells are the choanocytes. The examples of the poriferans include cycon, there is Scypha, spongilla, which is a freshwater sponge, and euspongia, that is the bath sponge. So in this module, we had carried out a detailed discussion on the different phylums, the criteria on which the classification is based, and discussed about the phylum Porifera, the unique features of the phylum Porifera. Thank you.